Okay, welcome everyone. Hello around the world, and welcome to this week's Oxford Centre for Tropical Forests seminar. And it gives me great pleasure to welcome you to this week's talk, which is by Professor Richard Corlett, who Richard is from the Xishuangbanna Tropical Botanical Garden at the Chinese Academy of Sciences, and he was former head of the Centre for Integrative Conservation at that centre. Richard did his first degree in botany at the University of Cambridge and his PhD in plant ecology at Australian National University with extensive fieldwork in Papua New Guinea. And ever since then, he has lived and worked in the Asian tropics at Chiang Mai University, at the National University of Singapore, at the University of Hong Kong, and then since 2012 at the Xishuangbanna Tropical Botanical Garden. And his research focuses on the ecology and conservation of tropical East Asia, and he was the lead author for the Asia chapter of the 2014 Working Group 2 IPCC report. But he's written several books, and I think many people regard him as the foremost authority on the ecology of East Asian and Southeast Asian forests. And he's written a number of books, including The Ecology of Tropical East Asia, which was published in 2019. So I have always enjoyed Richard's talks and his books as well for his very rounded and broad perspective on the ecology and future of forests and the range of disciplines and perspectives that he brings together in his analysis of the challenges that tropical forests face in general, and tropical Southeast Asian forests face in particular. So welcome, Richard, and over to you. Okay, Richard, are you there? We can see your slides, but we've lost your sound at the moment. We can't hear you at the moment. You're muted. So if you just, sometimes the mute button is sticky, but if you try your mute button, let me, actually it's maybe because you've locked out again. Try now, Richard. Try and unmute yourself now.
Oh, okay. Yes, I was locked out.
Okay.
Okay, can everyone hear me okay?
Yes, we can hear you fine now.
Okay, good afternoon, everyone. Thank you very much for the introduction, Yadvinder. It's 11 o'clock at night here. I'm speaking from the Xishuangbanna Tropical Botanical Garden of the Chinese Academy of Sciences, which is in southwest China. We're about 40 kilometers from the Laos border, 40 kilometers from the Myanmar border, and a couple of 100 kilometers from Vietnam. We have a 10 square kilometer garden and a big research center with about 150 scientists. So these are drone views of the botanical gardens for those of you who've not been here. My original title was going to be slightly longer, but my daughter, who's a recent Oxford graduate, thought it was too frivolous, so I cut it down to a more serious title. But I still think of it as Run, Forest, Run, what I'm talking about. The impacts of climate change on tropical forests are obviously immensely important, as everyone in the audience already knows, partly because of the potential adverse impacts on biodiversity, and also the possibility of large-scale climate carbon cycle feedbacks over the remainder of the century. Although warming is projected to be less in the tropics, and indeed has been less so far than in the temperate zone, large changes in rainfall may occur, And tropical species have been expected to have narrower climatic niches, though the evidence for this is mixed. The map on the left comes from a paper by Ken Feely, who I think is in the audience, and it shows the minimum distances between current climates and 2050 climates in the Amazon under climate change. And in the top left, it's only temperature. Top right is temperature and rainfall, and then C to F or various different patterns of land use change and land use permeability. But the obvious take-home message from this, looking at the scale, is that minimum distances to future similar climates, which is the distance that you'd have to move to keep up with climate change, is going to be hundreds of kilometers in the lowland tropics. We can make use of the present-day correlations between ecosystems, climate, and other environmental variables to predict future forest distributions. Ecosystem distribution modeling. There's a great example published recently in Diversity and Distributions for Tanzania, based on the National Forest Inventory Survey, which suggests a 50% loss of montane forests by 2085 and decreases in most other forest types. And large movements in the boundaries between different forest types. There's other possibilities using correlative distribution models. You can stack species distribution models if you have enough information on enough species, but you've got to be very careful with the assembly rules. You can't just allow every species to coexist. It's not going to occur. There's more sophisticated possibilities. But all these methods have the limitations that they can only predict long-term equilibrium distributions, which may take centuries to actually realize in practice. They can't incorporate rising carbon dioxide concentrations. They can't cope with novel climates, novel climate environment combinations, which we're expecting. And they can't incorporate realistic seed dispersal and establishment time lags. DGVMs, dynamic global vegetation models, are more mechanistic, based on our physiological understanding of plant growth, but as a result, more difficult to parameterize. They can predict transient states, not just the future equilibrium. They can include the impacts of rising carbon dioxide, and particularly important for the tropics, they can model fire as well. It's still difficult to incorporate realistic seed dispersal and establishment time lags, however. The example being shown here is for a recent paper from Simon Scheiter's group in Frankfurt, which used their DGVM, AGGVM2, to predict changes in vegetation, forest types between 2019 and 2099. This just shows a really crude biome classification, but there's a lot more information in these models than this. However, there are limitations with these DGVMs. This is very low resolution. Grid cells, 1 degree grid cells really are not much use when you are looking at future vegetation, which in Asia at least is already fragmented way below that sort of scale. A paper currently under review in Biogeosciences, possibly just accepted, ran the models just for the western half of the region with fixed present-day carbon dioxide and with rising carbon dioxide. And this sort of plus or minus carbon dioxide experiment showed that rising carbon dioxide promotes transitions from savanna to forest and from deciduous forest to evergreen forest that wouldn't occur otherwise. So these DGVMs have some properties that appear realistic, but like all models, well, most models, they incorporate a lot of what we know about plants, but they also get tweaked so that they match what we're expecting to happen in the future. AGGVM2 assumes that all functional types are already present in the local species pool. So there's no migration lag. If you've got a desert now and you've got tropical rainforest at the end of the century, there's no problem in most DGVMs because the plant functional types are just there, ready to grow. The only study in the tropics that I've seen which actually includes some dispersal limitation was a study in 2012 for Africa, which included no dispersal, which means within each grid cell from 2000 to 2100, no new plant functional types were allowed. Unlimited dispersal, when any plant functional type can reach every grid cell, and one grid cell dispersal. So only functional types present in adjacent grid cells could invade the site. And that's about 100 kilometers dispersal, so it's a lot. DGVMs typically model a small number of plant functional types for computational efficiency over large spatial scales. And they're not computationally efficient. They use a huge amount of time. And for that paper for the whole of tropical Asia at 1 degree resolution, there's months of computation in that. And it's very expensive in time to do experiments. Smaller scale studies suggest that ideally we want individual-based, spatially explicit models with species-specific parameterization, and that these would be a lot better. Currently, we don't have the data for this in the tropics, and if we did have the data, we don't have the computer power. But various approaches to overcoming these limitations are currently being developed by various groups around the world. However, shifts of hundreds of kilometers by complete ecosystems by 2050 or 2085, which are implied by these models, are obviously not realistic. Nobody actually believes this is going to happen. Keeping up with climate change does not necessarily require movement of species. Forests can adjust to climate change by physiological responses among the existing plant species, existing individual plants, I mean. This is usually called acclimation, but I think probably acclimation should only be used for responses which are adaptive. And if the physiological response is death, it's not acclimation, I don't think. Compositional shifts based on the existing local species pool. So the turnover of local plant species, you can get changes in the composition of species and functional types based on the local species pool. And then you can have actual range shifts of species. Physiological responses, acclimations, not my field. Crudely, one can see this as balancing the physiological costs of rising temperature and changes in rainfall with the physiological benefits of rising carbon dioxide concentrations. So a question of major interest in a whole lot of areas is, are the costs bigger than the benefits? Or will tropical forests continue to grow well for the remainder of the century? A very crude summary of a varied and rather frustrating literature. I think there is evidence for a useful capacity to acclimate to warming, but certainly it's only partial. So warming still has its costs. There is partial acclimation of respiration, of photosynthesis, and of other processes as well. However, there seems to be a very limited capacity to acclimate to drought. And the figure here is taken from a very recent paper in Global Change Biology. It shows hydraulic traits measured on plants in the longest running drought experiment in Northeast Brazil. And the blue is the control, red are the plants which have been subject to 15 years through fall exclusion. And you can see no evidence at all for acclimation of hydraulic traits. The net effect of all that, at least until relatively recently, was that the sort of benefits of carbon dioxide were winning out over the costs of adjusting to climate change, at least on the plot scale, leading to increases in above ground live biomass across the tropics in tropical forests. There is now recent papers, which I think some of the audience were involved in, providing evidence that this may be over in the Amazon, it may have reached maximum in Africa, and it's still not clear for Asia. Compositional shifts are evident across the tropics wherever we have plot data over several decades. It's not always clear what these compositional shifts actually result from, but in many cases, they're consistent with expected responses to observed climate change. That's really the best we can hope for. Climate change is not an experiment that we're running, at least we're not deliberately running it, and we don't have any control over it, and we've only got one experiment. So the best we can look at is, are the changes what we would expect? Probably the earliest good study was from Ghana, and this graphic shows the major changes, the red arrows, the habitat score, basically across plots in Ghana over 2 years, two decades of drought, the proportion of 10 centimeter DBH stems, which belong to species adapted to the drier end of the forest spectrum, has increased. The proportion of deciduous stems has increased. The proportion of non-pioneer light demanding stems has increased, and the proportion of shade-bearing stems has decreased. More recently, looking at plots across the range of rainfall in West Africa, from the wettest to the driest, we see pretty much the same patterns, but you can see that the biggest arrows are all for the driest plots. So the plots are arranged from the wettest to the driest, and it's the driest plots which show trees, a big decrease in evergreen trees, a decrease in shade bearers, an increase in non-pioneer light demanders. Interesting, this should occur particularly in the driest plots, not in the wetter plots. Similar trends have been reported from the Amazon, and in this study, you can see, if you look at the whole community across I think there's 106 plots, there's basically no change. But if we look at the change in the functional composition of the recruits, the proportion of drought-adapted species has increased, the potential size has decreased, and we see also changes in the mortality. So climate change impacts, as we expect, are most obvious in the recruitment and the mortality of tropical forests. It's not surprising that there's time lags in the responses. Trees are long-lived and sessile, so slow responses are expected. Also, if you're only looking at 10 centimeter DBH stems and above, there's a low annual turnover, under 2% in plots that I'm aware of. I think it may be higher in some. So even with large impacts on mortality and recruitment, compositional trends will lag changes in climates and will be slow. And you've also got to remember that the largest trees in tropical forests will be centuries old. I say can be, but in fact they usually are centuries old and cannot be quickly replaced. Lags in response may also be the result of limitations in the available species represented in the seed rain. There won't be everything in the seed rain. And as we saw earlier, in the tropical lowlands, potential source areas for trees adapted to future climates are often going to be hundreds of kilometers away. There have been very few observations of latitudinal range shifts in plants really anywhere on Earth, compared with numerous ones for animals. There are more altitudinal range shifts, but relatively few for the tropics. More evidence for altitudinal range shifts. Note also that some of the compositional shifts I mentioned earlier may reflect range shifts. It's generally the data's not available to tell whether the change in composition reflects just selection from the local species pool or whether new species have been added to the pool. In intact tropical forests, routine maximum seed dispersal distances, the sort of maximum distance you get in the lifetime of a tree, ranges from less than 10 meters for species dispersed by mechanical means or by ants. I can't think of any trees dispersed by ants, up to more than 10 kilometers for plants with tiny wind dispersed seeds, and some big trees do have those. And in my part of the world, at least, flying foxes, fruit pigeons, large hornbills, elephants can disperse plants potentially over those sorts of distances. But in practice, most seeds in the forest are being dispersed between about 10 meters and 1,000 meters. Large winged fruits in the canopy of trees and lianas, rodents on the ground, passerine birds, most primates, fruit bats, which usually don't swallow large seeds, but just carry them to a nearby feeding roost within 100 meters or so. A smaller number of species are dispersed further by large canopy birds, hornbills and fruit pigeons in my part of the world, fruit bats, but only for tiny seeds that they swallow. Many carnivores eat a lot of fruits and have large home ranges, many ungulates as well. And really long dispersal distances, I've already mentioned. In fragmented or deformated landscapes, which is all of them in tropical Asia, we don't have any really large intact forest landscapes. Dispersal distances will usually be less, though in fragmented areas they may actually be more because open country birds fly long distances. And in my part of the world, the animals I've crossed out, that is most of the dispersal agents for large fleshy fruits, have been hunted out of the majority of forests, even within protected areas. Movement velocities depend also on the time from seed to maturity, which can be several decades for large tropical trees. So the maximum distance that a species can move in a century is going to be the maximum seed dispersal distance multiplied by the number of generations per century to a first approximation. So in big tropical trees, maximum seed dispersal distances will generally be in the 50 meter to 1500 meter range, with perhaps 3 or 4 generations per century. Obviously, a lot of small trees have more generations per century. Well, small pioneer trees, small understory trees may have fewer. But you multiply those two together and you're getting potential dispersal distances in a century of less than 5 kilometers, which is the lower end of the expected range of climate velocities. So this suggests that range shifts will not play a major role in forest adjustment to climate change in the next few decades. There are going to be exceptions to this. Species with exceptional long-distance dispersal capacities, I've already mentioned. Species with range outliers in nearby refugia. In a wet forest, you may have dry forest on ridgetops. In a dry forest, you may have wet forest in the ravines. We rarely know this, so the local seed pool, the local species pool is rarely known. And in steep topography. So in steep topography, climate change velocities are typically low, and species don't need to move very far because climate gradients are very steep, temperature gradients in particular. So in steep topography, seed dispersal is likely to be important in intact forests, at least if the fauna is there. A factor I haven't mentioned is the lack of establishment opportunities and competition for limited establishment opportunities, which may also limit migration rates and may do so more than dispersal limitation. A recent paper in the Journal of Ecology, but it's Switzerland, it's not the tropics, and it's a modeling study. And there's really no practical way that I know of to study this in the field. So we actually don't know much about establishment limitation on the rate of species range shifts. Okay, I see Ken Feely's in the audience, so perhaps he could explain where he thinks range shifts are involved in this paper in the question time. I put a question mark because I'm not very clear what is happening here. What the paper shows is an analysis of more than 20 million plant collection and observation records over the last 40 years from 191 New World ecoregions. That in most ecoregions, the plants recorded recently have higher thermal optima than the plants recorded decades ago. And that's also true to a lesser extent for water requirements. But whether this involves compositional shifts, range shifts, something entirely different, I don't know. What about the paleo record? Climates have changed in the past. Tropical forests have been around, well, arguably almost since the Devonian, certainly since the late Cretaceous. The deep fossil record provides evidence for an extraordinary resilience of tropical forests. Basically, if you went from the paleo record, it just seems impossible. Nothing short of a dinosaur killer would get rid of tropical forests. There don't even seem to be major dips in diversity. However, the best studied climate events, the Paleocene-Eocene thermal maximum and the Eocene-Oligocene boundary, appear to have been considerably slower than what we're facing in the next few decades. I say appear because at that range we don't know if there were not periods of very rapid climate change. And this is the problem when looking at the paleo record. So we don't know if the rate of change in the past is relevant to understanding future problems. But also, obviously, forests were not fragmented, the fauna was intact, seed dispersal fauna was intact. So it's a very different situation. If we look at the last 20 million years since the last glacial maximum, we have now a lot of pollen records from the tropics, which give much higher resolution data. But it's important to remember when you look at a pollen record, like the one on the left, that most species aren't represented in these records. It's only a subset of the species in the forest. The temporal resolution is typically low. It can be as good as 50 years, but usually, particularly in the tropics, it's more than 100 years. The taxonomic resolution can be as low as family level or genus level, and only fairly occasionally at species level. And you don't really know where the pollen is. So the pollen that you're looking at on a slide on a microscope is all together on one slide, but it could have come from multiple different pollen source areas, and we don't know if the plants were growing together. So this example is from a lake on a peninsula north of Hainan Island. So this is basically the southernmost part of the Chinese mainland on the edge of the tropics. And there's a continuous record for 20,000 years, high resolution by pollen standards, 50 years in some parts. And if you look at this, there's a near complete turnover between over the last 20,000 years, with almost no species shared between 20,000 years ago and the present day, which is not surprising because that's, I'm not sure what they're estimating. I think around 6 degrees of warming, big rainfall changes as well. However, if you look in more detail, if you look at consecutive 50-year records, there's no rapid changes. Most changes are taking hundreds of years. You get spikes of individual species, but no obvious rapid changes in the vegetation. Several studies in what we think of as core tropical forest areas show surprisingly little change in pollen over various periods of the late Pleistocene. So this example is a lake in West Africa. And if you look at the middle of the 3 graphs, it shows the ratio of lowland to lower montane pollen over 90,000 years. And it's really remarkably constant. There are spikes, but no big changes. There are big changes near the tree line, a little difficult to interpret in the top graph, and in the regional forest cover in the bottom graph. But overall, remarkably little change in 90,000 years. And this is a Holocene record from Western Amazonia, and a mid-Holocene 1 to 2 degree warming period with reduced rainfall shows up clearly in the lake chemistry, but there's no associated vegetation change. There's spikes in 1 or 2 species, but that could just be chance, a tree falling into the lake. You really just don't know. If you look at this, that's spanning, I think, 7,000 years in the Holocene, really very, very little change. Okay, so to start to conclude, to summarize what I've said so far, the paleoecological record suggests that tropical forests have been highly resilient to environmental change in the past. Whatever time period we look at in the past, they appear to be extremely resilient. This resilience has now apparently been reduced by other human impacts, particularly hunting and fragmentation. Hunting because it removes seed dispersal agents, fragmentation because it creates barriers, and even in intact forests, it may not be enough to cope with the projected rates of climate change in the next few decades. Plant range shifts will probably be too slow to play a major role in the fate of tropical forests in the next 50 years. So basically the cavalry is not arriving. Tropical forests will adjust to the extent they can by the physiological responses and in situ compositional shifts that we're now witnessing across the tropics. And I think research-wise, and this is kind of coals to Newcastle since I know everybody's doing this, we need to focus on these in situ adjustments, which involve processes operating on scales from the molecular scale to hundreds of meters if we're looking at the seed dispersal pool, which are relatively easily observable, can be modeled in detail with species-specific parameterization once we've collected the data, and can potentially be manipulated experimentally. And we need to do this for major tropical biomes, and we need to do it across the major tropical biogeographical regions. So I just like to finish just to mention some of what we're doing at XTBG, which is relevant to this. When I say we, I'm talking about the forest ecology group, the climate change group, as well as a whole lot of other people. We have about 150 scientists at XTBG. And recently we've been working on our 5-year plan. As many of you probably know, China works on 5-year plans. We're coming to the end of one. We've got a plan for the next 5 years. One of the big ideas for the next 5 years is building on the existing Chinese Academy of Sciences Southeast Asia Biodiversity Research Institute, CACIBI, which is based at XTBG and involves several other Chinese Academy of Sciences institutions. And we're currently working in Myanmar, in the north, actually pretty much all over Myanmar, in Laos, and increasingly in Thailand, including constructing a new 20-hectare plot in the Kra Isthmus. And part of the plan of in the future is to create a 101-degree forest transect. 101 degrees basically going from northern Yunnan down through Xishuangbanna, Laos, Thailand, down to Paso, which I guess is 102 degrees. So there's already forest plots along this route. There's several canopy cranes. We've got 2. There's, I think, now multiple flux towers and mega plots with everything measured down to 2 cm dBH, and seedling plots and various other things. So I think for our part of the world, this could be the basis for sort of Southeast Asia's contribution to the pan-tropical effort. But we're a fairly small institution, so we welcome collaborators. So thank you. I think I better stop there. Judging from my, yes, I talked for about a bit over 45 minutes, I think.
Okay, thank you very much, Richard. Thank you for that wonderful overview and the narrative that you went from the modeling to the ecophysiological and site-specific studies to these broader questions of rate of change. If you'd like to stop sharing your screen, and I'd like to invite questions from the audience. As I mentioned at the start, the best approach is if you just flag in a one-liner or more if you wish in the chat, just that you want to talk or what you want to ask a question about, and then I'll try and curate them. And when I'll invite you to talk to Richard, to switch on your camera and your microphone and introduce yourself and where you're from. And part of the pleasures here is that we have an audience, I mean, from the entire planet here as well. It's quite nice to get a sense of that diversity of audience as well. So feel free to start pinging your questions into the chat box now, and I'll have a look at those. I'll just kick off while we're waiting for the first few questions to pop in. Richard, one of your, towards the end in terms of research needs, you talked about looking across different biogeographical regions and seeing what the differences are. And would you like to speculate on why we might expect differences between those regions? You mentioned dispersal syndrome, climatic history, many of these factors. Would you like to speculate on where you might see perhaps more resilience and more sensitivity across the tropics?
That's an extremely difficult question, as you know. A lot of papers include this in their speculation at the end, but it's speculation based on the fact that, say, Africa doesn't appear to be behaving like the Amazon, or Borneo doesn't seem to be the same as Africa. I don't think anyone's come up with a priori expectations based on what we know. Or if they have, they haven't been borne out by any data. So for instance, huge proportion of the biomass in Bornean tropical forests consists of dipterocarps, which have huge wind-dispersed seeds, which are moving tens of meters at the most. And maybe in the long term, this will have an effect, but will it have an effect over the next 50 years? I wouldn't predict so. So I am not sure that we can do that.
Okay, thank you. Could we have a question from Andrew Friend? Andrew, do you want to introduce yourself and pose your question?
Oh, hi, yes. Thank you for a really interesting talk. I'm really into developing individual-based dynamic global vegetation models, and as you know, you talked about we use these plant functional types, and I'd really like to move towards more species-level representations. And you mentioned that the biggest limitation to doing that is the lack of data on individual species in the tropics. So could you tell us a bit about what the specific parameters that we need and that we do have and that we don't have and what we should be looking for? And I can then start to think about what we need to put into these models to separate species out. What are the, I mean, I suspect you're thinking about wood density, these functional traits and things like this.
Yeah, I mean, a lot of these functional traits are really correlational, but with the DGVM, you want mechanistic traits, and wood density is a complex trait because wood's made up of vascular tissues, support tissues. It's not a single, there's no mechanistic link between wood density and responses to climate change. I mean, I think you just look at a standard DGVM like DGVM2 and look at the traits we have in the functional types, which are currently basically made up, and or in AGVM2 that's kind of allowed to generate, and we want to measure those on actual plants. But it's going to be things like I'm not sure I can answer that sensibly in a very short answer. If you email me about this, I do have ideas, but I think it would take me about 40 minutes to talk about it.
Okay, thank you.
Thank you. Thank you. Just to follow on from that, I don't have an answer to that question, but I will mention that one of the approaches we're taking to try and answer that question is coupling long-term forest inventories with functional trait data for the species. So in particular, recently, one of my team, David Bowman, has been looking at the Australian forest census data that goes back 50 or 60 years, where we've got functional trait data and data on which trees slow down during drought conditions, which trees do well. And by coupling those two, we can start seeing things like which hydraulic traits matter, whether photosynthetic traits, etc. So I haven't quite got the answer yet, but that's an approach I think that will help you get the answers that you need, Andrew.
Yeah, I mean, I think, obviously we need hydraulic traits, we need photosynthetic traits, but we probably also need below ground traits, which we know very little about. And I think because we're not very good on whole tree traits, because they're not easy to experiment with. So it's, I think the approach Yadvinder mentioned is probably the way to get at the traits we need to know.
Okay, thank you. Okay, there's actually I'll skip one because I think the next question may be one that Andrew is as suited to answer as Richard is. So Andrew Farlow, do you want to pose your question? And Andrew Friend, you may have a thought about this one as well. And I'll come back to Jennifer's one after that.
Okay. Thank you. I must admit my internet connection is not great. I'll do my best. There was a report that I was involved in the other day, the State of the World's Plants and Fungi from Kew Gardens. And one of the big highlight lines in the Yeah, and all the stuff in the media is, you know, 2/3 of plants risk of extinction. We're in an age of extinction. This was Alexander Antonelli's comment. And I was curious, the first part of the question is, you know, they have smaller stems than the trees. Are plants more at risk of climate change? So that's the first part. And the second thing is, my part of involvement is that was the medicinal plants. And one of the things we're trying to do is predict what's going to happen to all these plants in the future. So can we use some of these models to predict the impact on medical plant distribution in the future? That's GGVMs, or is that just too data intensive?
No, I mean, we certainly can do that, and people have. But as for predictions of, I mean, are they going to be wiped out by climate change? I very much doubt it. I think in the paleo record in the late Pleistocene, we've lost one plant species, Picea critchfieldii, which was abundant in the southeast United States and is now apparently extinct as a result of what is extinct, apparently as a result of climate change. I don't know of any other plant whose extinction can be attributed to climate change. That doesn't mean it isn't going to happen, and it doesn't mean that climate change won't contribute to the extinction of other things. And I think there'll be obvious threats to species which can't move. If you're growing on a limestone karst or an ultra basic outcrop, you can't just move a few hundred meters upslope or a few hundred kilometers north because there may not be a suitable substrate. And many plants, as you know, are edaphic specialists. I have a review with Kyle Tomlinson out in Trends in Ecology and Evolution that looks at this. And we basically conclude we couldn't actually work out what proportion of plants this applied to. I may be wandering off the question. I don't think 40% of plants are going to become extinct by the end of the century. And I don't think climate change is going to contribute to, be a major contributor to this. Unless it's,
I'll add a little thought. I mean, these are sort of point estimates at best. So, you know, the uncertainty in the number, and that's not really good in the media because they just latch onto a big number. So I'm just curious, it is quite a big number, isn't it? 40%. Thank you. Thank you.
Yeah, I mean, how many plants have become extinct in Europe in historical times? 2, 3? I mean, it's tough to kill a plant.
Okay, thank you. Andrew, did you want to comment on the potential for modeling at the individual species level?
Yeah, I mean, that's certainly. Oh, sorry, am I interrupting someone else? Yes, you certainly can do. I mean, species distribution models, yes.
Okay, Andrew, did you want to comment on that?
I mean, I haven't been involved with any modeling of medical plant distributions, but there's no real reason why any species or plant type couldn't be just put into the model and it interacts with everything else, and we push it into the future and see what its distribution is. I don't think that there's any strong data constraint on doing that particularly.
I mean, the problem, the data constraint tends to be dispersal again with this, because what you can do is push its climate niche into the future, but whether it can occupy where its climate niche is going to be in future depends on whether it can disperse there. So you can't just use species distribution models to push plants around the map. Plants don't move that easily.
Great. Okay, thank you. Okay, we'll move on to the question from Jennifer McElwain. Would you like to pose your question?
Yeah, sorry, I'll just turn on my video so I'm not rude.
I hope I cited one of your papers.
You did.
Sorry, my video won't turn on. I think there's just so many, so apologies for being rude and not turning it on.
No
Thank you for a really excellent talk. It's wonderful being in Ireland, attending a conference or a talk in Oxford with a speaker in China. It's so global. So I'm just wondering, I'm a paleobotanist. I'm just really interested, how can we help? So obviously the deep time paleobotany record has huge limitations, but we have the advantage of time. So we can track species, biomes through time. So if there was one thing you would like us to do, which would help you in your work, what would it be?
I think I would like, and this may be asking too much, high resolution records like we have, high temporal resolution records like we have for the last 20,000 years from other windows earlier in the Pleistocene or in the Pliocene. I think there are a few from the temperate zone where you can look at vegetation change on the sort of, perhaps not quite the decadal, but the century time scale 3 million years ago or 5 million years ago. Because these are plants we know, I mean, plants which are very similar to present day plants. And I think we could learn a lot from that. The problem with the period from the last glacial maximum to the present is it isn't replicated. You've only got one, you've got one sequence at every point. But it will be nice to compare past climate changes, ones which were perhaps different parameters changed or different rates of change. Is that going to be possible?
Yeah. I was even thinking deeper times, so times of, you know, I suppose pre our modern flora, but that would be possible as well. So thank you very much.
I think the problem with looking before our modern flora is that we get into sort of circular arguments about this. XTBG has a huge group that works on Miocene floras, mostly on macrofossils. And the Miocene here is not actually that different from the present day. So you can look, if you were dumped in a Miocene forest in Xishuangbanna, you wouldn't immediately notice you're in the Miocene, except by the animals, but not by the plants. But going back much further than that, and it gets really difficult to interpret what things are doing.
Actually, I'll just follow up on that question. This is not as much a question to Jennifer, I think. Much of the examples that we're focusing on are previous climates that are warm or warmer than the present, and there are good reasons to explore that. But in the glacial periods, we know that at higher latitudes had strong fluctuations and rapid fluctuations in temperature, even from a colder state to warmer states than the present. But firstly, do we have evidence that there was similar rapidity of fluctuation in the tropics? And if there is evidence, can these more recent glacial periods tell us anything about the rate at which plants adapt to change?
Well, I don't know whether I can answer that. So I suppose my experience is the deep geological record. So for the cold to warm transitions, or the warm to cold in Quaternary, I haven't got the experience, but way back in the deep geological time, I suppose if we use extinction of species as a metric, it's not good. But if we use relative abundance change, then we see really profound changes in the fossil record. But extinction of species is not a good metric for us to monitor ecological response to climate change. But a relative abundance distribution, you really find rapid change alongside rapid climate change. So rapid biological response to climate change.
When you say rapid, Jennifer, I always think deep time paleoecologist, rapid means a few million years.
Rapid, a few 100,000 years.
In South China, in tropical South China, you can see some of these sort of end last glaciation, rapid fluctuations. You can see a sort of younger Dryas signal at some paleo sites, but none of the records seem to show very large changes. I think in many tropical sites, even where you've got high resolution records, you don't see evidence of these rapid changes.
Okay, before we move on to the next questions, I want to pick up on a point that Richard raised in this talk, and I see that both Ken Feely and Belen Fadrique are on this call, and I'm going to put you two on one of you two on the spot and say the data that you're showing from South America of thermophilization, how do you interpret that in terms of the relative importance of range shifts or compositional demographic turnover in trying to interpret that? What's your hunch about those data?
Yeah, well, first, thanks, Richard, for a great talk. And I mean, I'll try to answer some of that, Yadvinder, but Belen can jump in, and also Daniel Zuleta, who is another co-author on some of the papers, is here, and maybe some of the others. I mean, what our group we've been trying to work in South America and throughout the neotropics for a long time, trying to see how these forests are responding to climate change. And I mean, the consistent results that we always get is that the composition of the forests are changing to include more heat-loving species, but those changes are slower than required to keep up with climate change. And in almost all the cases, those changes appear to be driven primarily through range contractions of the species rather than range shifts or range expansions, right? So basically, species appear to be primarily, migrating. I mean, we still call it migration, but it's kind of a funny term. They're migrating by dying out from the areas that get too hot for them, and they're only slowly, if at all, moving into areas that are becoming newly suitable for them. Why that is, right? There might just be this lag that things obviously can recruit slower than they can die, or there might be competition that's keeping them out of those areas, or there might be edaphic conditions that keep them out of those areas, or other climatic factors. We don't really know yet. But it seems to be pretty consistent that for the vast majority of species, ranges are contracting through time rather than actually moving. So if you want to call that migration, you can, but it's a migration kind of in the worst possible way.
Thank you.
Interesting.
Okay, great. Okay, so let's move on to some of the other questions. So Shyam Patel, do you want to pose your question?
Thank you. It's a nice talk. So I'm wondering, do we have enough regenerations or seed traits except the seed mass? Because the seed mass is one of the soft traits, seed mass and seed shape, or little bit dispersal traits we have. Do we have hard traits like seed dormancy, seed germinations requirement, and the seedling banks, this kind of the things? So about the tropical forest and to tell about their shift range. Because I being a seed ecologist, I found these are the least studied area in a tropical forest, except a few from the Latin America.
I mean, that's an interesting question. Our seed bank within the gardens, which is has done quite a lot looking at seed characteristics to some extent in relation to climate change. But I mean, I would agree with you. We don't have enough data, and we don't actually know. And I'm sure there must be someone else in the audience who could say more about this. If we get these high resolution, high spatial resolution models dealing with species-specific parameters, then seed characteristics can have to be included in that. And if we're looking at sort of time lags in establishment, or how invading species establish in competition with the existing canopy, then seed characteristics are going to be part of that. I'm afraid I can't really do more than confirm the fact that we don't really know.
Yeah, I personally feel we have a lot of functional traits, but it's still the hard seed traits are missing, not even in tropical, but even the temperate from biome, that we need to focus more. Because the climate is going to be deal differently to the adult plants, and then climate is going to deal differently with the seed, because seed is more sensitive for the climate change.
I would have to say that we don't actually know that. It may be that that germination is going to be massively affected by climate change, but we do not have the data to show this. So, which is why we ignore it and why it isn't in any of our models.
Thank you.
Okay, thank you. Okay, we'll move on to a question from Sabrina.
Hi, thank you for giving the talk. I think I need to apologize first. I think I'm going really late, so I need to ask, probably this is something you addressed already in the talk, is considering that I think the species move, so plant, you said not move really quickly. So is it still, I don't know, useful to do many climate-based projections that spans 50, hundreds of years, because lots of policies made in the couple of years even, or even not a decade. So how do you think we can address this in the policy?
Policies certainly are made by and for people who tend to be only looking a short while ahead. But policies that relate to forest protection and facilitating adjustment to climate change need to look at both short and long time scales. So obviously we need to worry about what's going to happen by 2050. That's a period which most of you are going to see. And but on the other hand, the biggest trees in the forest may be 500 years old, and we need to understand how these things are going to behave in the future. And basically, forests which are not protected in the next 5 or 10 years won't be. So I think this applies to ecosystems in general, that we, unless we make a commitment to protecting, say, 30% of ecosystems, or maybe even 50%, it's going to be too late, and we're going to have to instead work on restoring that much. I'm not sure I'm answering your question, but we have to work on multiple timescales, and I focused on this period because it's the period where there's information available. But I do think people should be looking 500 years ahead.
Okay. Following up on that, I'm not sure I get that you make the model specific to different biogeographic regions, because I think like different places have different species, history, community, composition, etc. Like how far into the local scale do you need to be really specific in those models actually?
Well, I'm suggesting, I mean, I think, and people are working on this, that we need to have study areas where we're looking at all these things, from the where we have 50-hectare plots plus knowledge of the surrounding areas, perhaps small plots scattered over a much wider area around them. We need canopy cranes, so we have access to the canopy. We need eddy flux covariance towers. We need, these are going to be different in different parts of the world. When I first started working in tropical Asia, tropical ecology was by far best developed in the near tropics. Really, Asia had its time earlier, that it was sort of the near tropical period. And what tended to happen in Asia is that we just said, well, this happens on Bawah Colorado, so it must happen in Borneo. We now know that's not true. And Richard Corlett and I have written a book on why this isn't true, and it isn't true. But it's possible that some things will be the same pantropically. We don't know, so we need to look at everywhere.
Okay, thank you.
Thank you. We'll move on to a question from Ollie. Ollie, would you like to pose your question?
Hi, yeah, thanks. Can you hear me okay?
Yep.
Yeah, thanks for this. Yeah, I was really struck by your two slides on dispersal, on seed dispersal, and then you come to the one with all of the vectors crossed out. And then it seemed to me that the largest potential for dispersal left on your crossed out list is human. And given that obviously anthropogenic factors are so huge in shaping all of the outcomes, then, and also somehow seem to sort of, in some ways, maybe break the mold in terms of the generation model, because obviously it doesn't necessarily rely on that and they move in different ways and over different distances. I mean, if you had to speculate, how significant do you think those factors are going to be? And, you know, are they going to be differently significant in different areas?
Which factors?
The human-led dispersal.
I mean, in tropical forests, unless we actually decide to do it, very few species, I can't think of any, there must be some, are dispersed sort of accidentally by people. But there are huge forest restoration projects planned or in operation, like in the Brazilian Atlantic forest area. There's probably several thousand forest restoration projects, mostly small plots in tropical Asia. So tropical forest restoration provides an opportunity for assisted migration, of moving the species into their future, into suitable future climates. So yes, if restoration, particularly planting, is stepped up, then it could be hugely important. But it's not going to happen accidentally. I don't think weeds get transported on people's vehicles and in people's luggage, invasive species, but not dipterocarps or understory rainforest trees.
Okay, thank you, Ollie. Okay, it's getting late, so I think I'll end with one final question, which is a little bit of a tough one, but it's a more philosophical one. And as you've highlighted in your talk, Richard, the rate of change is one of the key features of contemporary climate change. It's the speed at which we're approaching a 3-degree world or 4-degree world, and a variety of scenarios. And you know, as a research scientist, we always want to do more research. There's more we need to understand about the system before we can be confident about predictions or interventions of the system. And a real challenge I think we all face as environmental sciences is what this rate of knowledge accumulation and understanding is compared to the rate of change in the wider environment in this context. And within that, certainly need to reconcile what are the low-hanging fruit, what are the priorities where we're likely to have a tangible progress and understanding in the next decade, say, that can actually be tangible in terms of interventions and conservation in the next few decades. So I'd just like to pose to you, or to anybody else in the audience, how do we reconcile that? How does that help us prioritize the type of science we do and identifying the more tractable problems rather than perhaps the more interesting but more intractable problems that we tend to often be attracted towards as scientists?
I'm a slow thinker, particularly after midnight. It's a difficult question. I'm not sure there are any low-hanging fruit. Having worked in various bits of ecology and conservation biology, I think conservation biologists often think, well, maybe we should stop doing research and maybe we should just go out and fix everything, or get other people to fix it. And put our time into public education. And I don't think that's true for understanding the impacts of climate change on tropical forests. I think we're really knowledge limited. And one of the things put in this talk together has made me realize is how many gaps there are, and there's gaps everywhere. I'm not sure there are low-hanging fruit, but I would certainly, the low-hanging fruit were existing plot data going back 20, 30, 40, 50 years, of which there was a lot. And I think a lot of that has already been looked at. And I think perhaps if I had large amounts of money, I would maybe putting it into large-scale experiments, sort of forest-scale warming experiments replicated across the tropics, carbon dioxide enrichment. These are going to be a real pain to do, but I think until we do them, we've learned a lot from the through-fall exclusion experiments, despite the fact they don't mimic real droughts terribly well, but they have taught us a lot. And some surprising stuff. I think experiments are valuable and underutilized. We've tried to find experiments that have already been done, and then we can go and look at the results.
Okay, thank you. And I know it's getting very late in China, so I think it's a good time to wrap up. But as Jennifer commented, I think it is fantastic that we have these events and we have comments and perspectives coming in from South America, from India, from China, from Europe, elsewhere. And I think it certainly is in one of the bizarre ways in which the pandemic is increasing connectivity and open also democratizing it in the sense that you don't have to be able to travel to a conference to be able to access thoughts and ideas. So thank you for giving your time and commitments to this effort. As I mentioned, but this is being recorded online and we'll share it on YouTube, and feel free to share it with your students and colleagues. So
YouTube's not accessible in China.
Sorry about that. If there are other formats that you think actually would be better for China, we could also upload it into.
I will ask my students.
Yeah, just send me an email and I'd be happy to share it in other formats as well. Great. Thank you, everyone. Good evening. Good night.
Bye, everybody. Good night.